Welcome to Biology by Bradford. If you're a student, I'm going to take the big, scary looking stuff and break it down into pieces that actually click. Today, we're going to look at inheritance. You carry two copies of almost every gene, just two, and that one fact decides whether a condition shows up in every single generation of a family or slips silently through people who never know they're carrying it. Today, we'll figure out which it is. Now, I've got a presentation that I would typically give in class, and I've jumped to this page already. And then I'm also going to show you one of my HTML interactive web sheets where you can use it to review yourself. Now, autosomes are all the chromosomes in our body that do not determine the sex. So we've got 23 pairs of chromosomes, 22 of them are autosomal chromosomes, and then one of them is our sex chromosomes. And we're going to look at autosomal inheritance. It's the easiest place for us to start. Now, here is a nice mind map giving you an overview of everything. And what I'd like to draw our attention to is this idea that we've got all these different terms we need to get our head around. Homozygous recessive, homozygous dominant, heterozygous carrier, autosomal, heterosomal. Now, for my students, I always ask them to use Cramly, which is an app that I built for spaced repetition. Because if you can just memorize and lock in that jargon, then the conversations you're having in class and the explanations I'm giving now is so much more accessible. Because let's face it, these words are nonsensical. They're not ones that we use every day in a conversation. I don't go and talk to my mates down the pub about a heterozygous or carrier or any of that stuff. Once work is done, I'm out of the door and biology is done for the day, typically. So I really recommend spaced repetition. It's a really efficient way to learn, and check out cramly.study, and I'll share the link below. So we'll start with what is a gene. So it's a heritable factor that controls a specific characteristic. Now, for example, we always use, as biology teachers, eye color. However, it's a little bit of an oversimplification. It's much more complicated than that. But it's easy to think, there is an element of if something is dominant or recessive. And then we know that brown eyes is dominant over blue eyes. Both my parents have dark brown eyes, but you'll notice that I have blue eyes. Why is that? It means they were both carrying the blue-eyed gene. And therefore, they must have been heterozygous. And the dominant gene was being expressed in them. And what's interesting is the recessive one is also expressed, but it is like dominated. So there is an expression of it, but whatever characteristic phenotypically of the dominant gene is overriding and it masks the effect. So that's when we say that someone is a carrier or the dominant gene is masking the effect. We literally mean it like putting a mask on. Although it's being expressed, whatever the dominant gene is expressing hides that. Now, it's really worth checking out some videos on Mendel, especially when you go into dihybrid crosses. He's the godfather of genetics. He helped us write the rules on it. But for today's lesson, we don't really need to dive too much into what Mendel did. We just need to appreciate that on every chromosome, we can divide different sections into genes. They are heritable, and variations of a gene is called an allele. Now, I'm old, so I had cassette tapes. You can think of a cassette tape as a chromosome. And then one song on that chromosome or on that tape is a gene. And the tape itself would be like our DNA. And we could even say that being coiled up around here is like being wrapped around a protein, because we know that eukaryotic DNA is associated with proteins. And different variations of a song, like a remix, would be an allele. The physical expression of something is what we call the phenotype. So tongue rolling, not tongue rolling. It's a nice one for that you can have a look at. It's very visible. You can either roll your tongue or you can't roll your tongue. And it's controlled by a gene. I can't roll my tongue. And it's not something I'll ever be able to learn to do. So that is a physical characteristic. Now, when we're trying to figure out what is the possibility of inheritance, we always use something called a Punnett square. And a Punnett square will take, and it's easy to use flowers and stuff with this, it will take those like genotypes of the parent. So genotype is like big R, little r, or big R, little r. And it will then separate them into the gametes because you only inherit one copy from each parent. But there's a possibility of inheriting big R or little r from this parent. So if we sketch this out into a Punnett square, and this is where I'm going to flick over to my web sheet in a minute because it's interactive and I like it. So here's the egg cells, big R or little r, the pollen, big R, little r. And we end up with a 3-to-1 ratio. Now we would actually write these round the other way. You always start with the capitalized one, but it's just to demonstrate where this is coming from to make that more uniform. But you've got a 3-to-1 ratio of phenotype, and that's a number that needs to stick because each one of these here would have the same expression. It would express the dominant gene. And these two here are being masked. They're carriers of that recessive gene. That might mean absolutely nothing. It might just be to do with eye color, or it could be an inherited disease. So I just skip down. This is my web sheet for this one. I'm not going to talk through it. It's something for you for your reference. And we've already looked at this. But what I think is really nice, if we skip down, is where we can build our own Punnett squares. So I can change the parental ones go in. You can see we got our ratios down here. If I switch that up and make these two big A, what effect does that have to the phenotype ratio? Well, now they're all dominant, but we have this 1-to-1 genotype ratio, 0% recessive phenotype. Of course, if I make it like this, they're all going to be homozygous recessive. So you can have a little play around with that, and you can see the different combinations. The main ones would be this 3-to-1 ratio of phenotype, or in something that's called a test cross. Like if I don't know if this one parent A is like this or like this, then I can do a test cross. So let's see what happens. This is a test cross. I can figure out now because they all end up the same, heterozygous. I don't know they're heterozygous. I can assume this because they all end up 100% having the dominant phenotype ratio. And there is no homozygous recessive one expressed. So I know that it's going to be homozygous dominant. If I was doing a test cross, always with a homozygous recessive, like this, then I end up with a 1-to-1 ratio. Now, I now know the individual was heterozygous for that trait. The two that we want to learn about in IB, the two examples, would be cystic fibrosis, which is homozygous recessive, and they're both going to have the same crosses. So cystic fibrosis is something that affects a few different parts of the body, like the production of digestive enzymes. One of the biggest things is like the clogging of your alveoli and clogging with mucus, and you need to have lots of like cupping physiotherapy to help dislodge this. But actually, the way that medicine's progressed, if you're diagnosed with this as a child now, you end up with like living a normal, healthy life. My friend was actually diagnosed with this at like 5 or 6. Her parents were told that she was going to live to 12, and she just celebrated her 39th birthday. And that's because of the medical advancements that's happened in conjunction with her progressing in age. So she's now on something called a Cambi, which I've got linked here, and it's a drug that like literally like she has not been in hospital with a chest infection since this drug, and it's quite amazing. Both her parents don't have cystic fibrosis, so that means that they were carriers. So like this cross here, they were carriers, big A, big A, little a, and she had a 1 in 4 chance, a 25% chance of having cystic fibrosis. Every time a new offspring is conceived, the chances are the same again. So her sister doesn't have it, but she didn't have like an increased chance because her sister had it. It was still the same because it's a lottery every time. And we've also got something called PKU. So this is a autosomal recessive condition as well. This is something that you get screened for as a baby, and if untreated, is really damaging and can end up with serious mental retardation. But if you figure out that someone has this recessive gene and is homozygous recessive for it as a baby, then you will live a perfectly normal life, and they will be able to manage your diet. And the other condition, though, is Huntington's disease. So Huntington's disease is autosomal dominant. It's something that still exists because it's something that doesn't express itself fully until you are past that reproductive age. So Huntington's disease would be like, in this case here, with two heterozygous individuals, this would be the person that doesn't have it. This comes in to more into play when we look at pedigree charts in the following video. So I'll just sign off by saying, check out this web sheet. And at the end, I've always got, if you skip all the way down, I've always got some multiple choice questions here, on long answer questions that you have the mark scheme so you can practice, which would be really good for any little assessments. And the one thing to lock in is the terminology. So check out Cranby for that. And yeah, I hope you've enjoyed today's video. Please leave any comments, like, subscribe, share, any feedback, always open to it. I think that's one of the most important things as teachers and as individuals in life, to just be able to take on board feedback to improve, because I want this to be useful for as many people as possible. So, thanks for today and catch you later. Ciao.